Thank you, everyone. Thanks for joining. Welcome to the session by SGS Metrology. So I will just start with a general overview of SGS, how it can help accelerate the release of pharma and biopharma products, and then also we'll focus on the Centre of Excellence for Biosafety Services, for cell banks, viral seeds, and gene and cell therapies and viral vaccines. And then we'll go on and we'll show a case study where that Centre of Excellence, together with other parts of the SGS CRO network, were able to deliver more than 30% of the vaccine for COVID-19 by the end of 21 for the whole world population. So SGS businesses, there's 5 business lines. They test many different things from minerals, oil and gas, agriculture. They certify shipments, etc. So where we fit is in this health and nutrition sector, which is involved in the testing of biopharma, pharmaceutical products, and also food. And there's also a clinical trial section within that business line. So why work with SGS? So we are experts in a wide range of innovative and integrated analytical testing solutions, clinical research for both small pharma molecules and large biopharma molecules, including advanced therapeutic medicines such as the more complicated viral vectors and also viral vaccines. And in this way, we are able to help reach global markets very quickly and ensure safety of the product. We have a very integrated CRO network across the world that use state-of-the-art facilities and a wide range of different scientific experts in many different disciplines that can take all the molecules from the early phase of drug discovery right through to clinical trials, batch testing of clinical trial material, and then eventually the commercialization of that particular drug product from a wide range of different diseases. And also we have a global regulatory compliance harmonised together where the entire network works to the same compliance from a GMP, GLP perspective. And we're very customer-centric. We are very good at delivering for our customers, on-time delivery, meeting the needs and trying to exceed the expectations of our clients. And that is our main focus in this industry. So this really just gives you a list here of the different sites throughout the SGS Health Science Network that are focused on either biopharma or pharma, or sometimes both. And there's also a CDMO in the pharma industry in the United Kingdom in Deeside called SGSK Pharma, which was a recent acquisition. So we'll focus really on the Glasgow site and the services here and how it fits with the biotherapies and what it does. And you'll hear from my colleagues Richard and Ewan later in more detail about the specific methodologies that we use on the Glasgow Centre of Excellence site. This really just shows the different types of capabilities in North America, Europe, and Asia Pacific. And the ambition of SGS is really to have this type of capability on each of those 3 regions to deliver excellence within those regions for all of the services that we offer in the industry. So what kind of analysis do we do for biological medicines? So, for example, we do the characterization. We're then involved in the quality of the actual batches and the safety, and also in the potency of the batches. And we're involved in a lot of our batch release testing. Many sites are very focused on the batch release testing to allow rapid access into the patients. And we support the production of all biomedicines and in the global arena. So you can see we can do protein characterization, cell bank that uses the that's used in the production system, and the virus that may be used, or the virus vector, vaccine identity testing. We can test raw materials, characterise the raw materials, and also look at purity and process impurity tests to ensure that the limits are not above the specification. And classical sterility, endotoxin, and the site, this is more the speciality of the Glasgow site in the safety arena here: sterility, endotoxin, virus detection, and microbial and adventitious agents, and together with the residual impurities. And we can also offer potency testing on drug product using cell-based and in vivo-based platforms that would be outsourced. So this is a diagram here of the Glasgow site, which is about 3,100 square metres, with ambitions to more than double that size by 2025. So we test cell banks and the raw materials for growing the cell banks that are either used for the bioproduction of the viral vectors or vaccines, or they may be used to express the recombinant therapeutic, wide range of different types: CHO biologics, microbial, SF9, HEC293, etc. And then also the harvest from the bioreactor, and then the downstream final products, whether it be a recombinant protein or vector or cell therapy or whatever other type of biologic. And also the seeds of viral vaccines and vectors that are used in the cell bank culture and the bioreactor and the harvest of those. And also cell therapies, which can be modified or not modified depending on, on what the therapy is and their actual derived products and drug product. And then this is how we actually support all of their global clients in the manufacture of the different biologic therapies which are named here. So just some background on the team and the methodologies that you'll hear Richard and Ewan going into. So it's a specialised team that was set up back in 1996 in a company called Q1 Biotech in Glasgow. And it was employed really to deliver new virus detection system, and this team pioneered the development of real-time PCR and PCR, nucleic acid sequencing, electron microscopy, and cell culture-based assays in the early birth of the biologic medicines in those days, and viral vaccines under GLP and GMP compliance. Very unique site. And then in 2007, there was a team of 17 that started up what was termed Vitrology, which is now SGS Vitrology or SGS Glasgow on the market. Vitrology now being the brand name within the SGS global arena. And the same team still remains today, which shows the robustness of the facility and the compliance. We've been inspected many times by MHRA, FDA inspected since 2008, and we were involved in 2009 in the emergency response for swine flu pandemic to release hundreds of millions of doses in less than 6 months. We joined SGS in 2012, and we've had a multi-phase, multi-million pound exponential investments over the past decade to reach almost 150 team on site with about 3,000 square metres and a wide range of methodologies. In 2020, we executed with the Oxford University and clinical biomanufacturing unit there, and then subsequently AstraZeneca, once they took the license, to develop the first batches and deliver over 3 billion doses of AstraZeneca COVID-19 vaccine called AZD122 and Covishield in India. Today, we have over 500 global biopharma clients, and our on-time delivery for batch release and testing is around the 90% mark. Going forward, we expect to be launching another multi-phase investment, multi-phase facility to reach more than 7,000 square metres and 300 FTEs on the Glasgow site. And we release all viral vaccines and biological medicines that are made throughout the world, including these ones here that are listed. This really just is it. You hear from Richard and you in more detail, but this slide is really just for information purposes about how the types of tests that we offer out of the site for the different phases of the bioproduction, the raw material, the cell banks, etc., the unprocessed bulks, and then on the purified drug substances. This is the case study here that really just shows how we created with Oxford the risk assessment to deliver the first 500 doses in March of AZD122 vaccine using novel molecular biology methodologies, fully validated, fully GMP. And then we committed a huge investment to ensure that that was a front runner on the race to market for COVID-19 vaccination of the world population. By June, we had delivered the clinical batch testing and the clinical trials were well underway. And we then helped AstraZeneca Oxford get the whole thing through the MHRA, validation of all methods, and the production systems, etc. And then subsequently the approval of AZD122 in over 180 countries in early 21. This then gave a huge worldwide demand for AZD122, so we had to invest further and build a 650 square metre facility, hire FTE and train them at pace within short months to deliver the capacity for the world. And by the end of 2021, we had released over 3 billion doses with between SGS Glasgow, Wavre and Lincolnshire. And the CDMO network shipped all the samples into the SGS Glasgow and Wavre sites for using this diagram here. Excuse me. This diagram just shows how manufacturing of COVID vaccine with the ChAdOx vector, master seeds, working seeds, cell banks, the bioreactor, the control cells and the purified drugs. And it was a 56-day turnaround using the longest duration test on that drug product. And that's the type of testing here that was done on viral bulks, control cells for adventitious agent. Richard will go into more of this. And then on drug substance, a lot of the sort of drug substance release and drug product release testing that we offer. So thanks very much. And sorry about my cough, but that's me, and I'll hand you over to Richard now. Thanks.
Thank you very much for the invitation to speak today. So I'm going to talk about sort of considerations for biosafety testing on cell and gene therapies, and looking at particularly virus contamination risks, and specifically focus on the virology assays. I was asked to focus on the in vitro assays. So this is a sort of an overview, a schematic of the biosafety testing of cell and gene therapies. So if we concentrate on the left-hand side of the diagram in green for gene therapy, where we have a viral vector, so this is going to be put directly into our patient. So that viral vector is going to have to be produced in a cell bank. So we're going to follow the same principles as any cell bank for any biologic, master cell bank, working cell bank, and a production cell bank, and testing of the viral vector. We're going to need the complete testing package of those materials, and I'm going to talk about those in the upcoming slides. I'm just going to introduce here the concept of control cells. So this is where we may have issues being able to test the viral vector. So for example, if there's toxicity that we can't neutralize, testing the control cells is an option. On the right-hand side of the diagram is an example of a cell therapy, and most cell therapies these days are a combination of cell and gene therapy. So here is an example of CAR T therapy, where we will extract cells from our patient, and we then have a very restricted time frame in which to transduce those cells with our viral vector, usually replication incompetent lentiviral vectors for CAR T therapy. But because we have a matter of days here, we don't have time to do the longer duration assays, which I'm going to show you in the upcoming slides. Here, we're much more restricted to the PCR-based rapid methods. However, because the viral vector that is used for the transduction is still viewed as final product because it is so important to the process, then you still have to do all the testing on the left-hand side of the diagram to produce the viral vector. So this is a sort of schematic of the testing that is required for cell banks, virus seeds, and vectors. The manufacturers are going to have to prove identity. They're going to have to prove genetic stability and purity at each stage. So that's basically freedom from bacteria and fungus, from mycoplasmas, and also from contaminating viruses. And virus detection, which is going to form the basis, the bulk of this talk, falls into three main areas. So firstly, we have our generalized assays, adventitious agent tests, or what we call our in vitro assays, that are not looking for anything specific. So they're looking for the evidence of anything infectious. Because retroviruses, when they are associated with human disease, tend to be associated with severe disease, it is a regulatory requirement to look for the evidence of infectious retrovirus. And then thirdly, depending on the cell line and the product and the species, there will be a species-specific approach. So for example, if you've got a human cell bank, you might have a human PCR package to look for the most likely human virus contaminants. So I'm going to show a quick slide on the identity, genetic stability, and purity, and then focus mainly on the virus testing. So for identity, there are several ways of doing this. And one of the most common ones we use is on the right-hand side here, the rapid amplified polymorphic DNA. This is essentially a low stringency PCR with 6 arbitrary primers, and you can compare the sequence to a reference standard. We can also use Sanger sequencing and also next generation sequencing to prove identity. In terms of genetic stability, the take-home message from this slide really is that what you're trying to show is comparing your starting material, your master cell bank, to your end of production material to show that there's been no gross genetic changes. And there are different ways you can do this, such as transgene copy number and also structural analysis using southern blotting. In terms of purity, freedom from bacteria, fungus, sterility, etc., there are broth methods that are well established. However, that's on the left-hand side of the diagram, if you remember. On the right-hand side with the CAR-T therapy, we're going to be restricted to the rapid PCR-based method. So this is where we have things like the PCR MicroSeq. Virus safety really falls into 3 main areas. Firstly, selection and control of the starting materials, and then thorough testing of those materials. And thirdly, processes, the downstream processing to remove potential virus contaminants or viral clearance. However, it's important to realize that for viral vaccines and gene therapies, there is little to no viral clearance, and therefore the theoretical risk of contamination is higher. So there's much more emphasis then on selection and testing of those products. When we're thinking about a risk assessment, again, if we think back to our cell bank, if our cells have ever been exposed to bovine serum or porcine trypsin, even if they're now free of those materials, we have to consider they could be contaminated with a range of bovine or porcine viruses, and therefore is a regulatory requirement. This is described under the US Code of Federal Regulations, Chapter 9. So we call this testing 9 CFR, and it looks for a range of specific viruses. Depending again on your cell material, there may be specific risks. So I've given an example here of MMV, which is a known contaminant of CHOs. So if you are manufacturing in CHOs, you may look specifically for MMV contamination. On the top right of the diagram, again, if this is a manufacturing process, so if you get a breakdown in manufacturing control, you could get operators, for example, introducing contaminants. However, from a virology point of view, we're very concerned about dangers posed by latent viruses such as herpes viruses and endogenous viruses such as retroviruses. So how are viruses detected? So the take-home message today is it's a holistic method. There is no one method that will detect all viruses. So it has to be a holistic testing approach. On the right-hand side of the diagram, that I'm not really going to talk about today just in terms of time restrictions, we can look for markers of infection. So we can look for direct detection of particles by electron microscopy, and Ewan's going to talk about that in the subsequent talk. We can look for virus genomes by PCR, PCR-based methods, or we can look for the evidence of virus proteins, things like reverse transcriptase activity, indicative of a possible retrovirus contaminant. On the left-hand side, if you want to look For infectious virus, though, you have to use a culture system. So in cell culture, we look for things like cytopathic effect, and I'm going to show you some pictures of what we mean by cytopathic effect in the upcoming slides. If you want to look for pathology, then you have to look in animal systems. So this is a schematic of our 14 or 28-day in vitro assays. Typically, you do 28-day assays on master cell banks and the 14 days on the bulks. Essentially, what we will do is seed our cells the day before inoculation and then prepare a lysate from our client's material and inoculate our cell monolayers, and then observe them over 28 days for cytopathic effect. The standard endpoints are cytopathic effect, CP, and hemoabsorption. And again, depending on which regulations you follow, you may also include a hemoagglutination endpoint. If you combine the regulations together, the requirements, you come up with a minimum of 3 detector cell lines. So we use a human diploid cell line, MRC5 cells. We use a simian cell line, for example, Vero cells, and a third cell line that is as close to the cell line of manufacture. So if you're manufacturing CHOs, you use a CHO indicator cell line. The take-home message from this slide really is why we include MRC and Vero cells. So you can see that both of those cell lines support replication of a wide range of human viruses. So this is an example of CP. I'm going to show you a few pictures. So in all our assays, we have negative controls that are mock inoculated to control for the health of the cells. And in a separate laboratory, we have the same cells that are purposefully infected with a range of model viruses. And that is to give us reassurance that the cells we're using to detect a contaminant can support replication of a model virus. So what I'm looking for as a scientist is that the CP appears in the time frame I expect, and it develops with the morphology that I expect. So this is an example of adeno-CP about 5 to 7 days post-infection in Vero cells, forming a nice rocket, and within a few days that will destroy that monolayer. This is characteristic of HSV1 CPE. So on the right-hand diagram, the leftmost arrow shows some swollen rounded cells. So this is very common of HSV1. And the middle arrow points to cells that are fusing together called syncytia. So this is very characteristic of HSV1. Again, another example here of measles-infected Vero cells. So you get these multinucleated giant cells. Some CP is very easy to read. So this is EMC-infected BHK21 cells, only 24 hours after infection. And we use this a lot for training operators because it's safe to use and it's easy to read down the microscope. Many viruses, however, don't produce CP. So one of the other standard endpoints is to look for hemoabsorption. So this is where we will perform our assay for 28 days, do a CP read, remove the medium, wash them with PBS, add a solution of red blood cells, incubate them for 30 minutes at either cold or ambient temperatures, then remove the excess blood and read them for CP there for hemoabsorption. And this again is a positive control. So here, what you can see arrowed are small clusters of guinea pig red blood cells tightly bound to the surface of infected MRC5 cells. And these are very tightly bound, so even if you bash the flask, these little clusters of bound red blood cells won't attach. So if we saw this with a client material down the microscope, we'd be very concerned there was a hemoabsorbing virus in there. Not all viruses produce CPE or hemoabsorption, so one of the other endpoints required under the FDA vaccine guidelines, and again under potential consultation with the new version of ICH-Q5A, is the hemoagglutination endpoint. So in simple terms, if you take red blood cells and add them to the bottom, add them to a V-shaped well, by gravity they will fall to the bottom. And after about 30 to 45 minutes, when you look above from a light box, you'll see a very nice tight pellet being formed. However, if you have a hemagglutinating agent, such as an influenza virus in there, it will bind all the red blood cells together and then it coats the surface of the well. So when you look from above, you see a very diffuse pattern, and you can see that in the HA positive wells there. Not all viruses produce CPE or hemoabsorption or hemagglutinates. So there are some viruses that we have to look for specifically. So if you remember back to the picture with the cow and the pig, the 9CFR testing, most of those viruses we have to look for using fluorescently tagged antibodies. In terms of detecting retroviruses, there are various ways of doing this. So you can look by PERT assay, and you can look by TEM. If you get an unexpected PERT positive result, a regulator will ask you to perform an infectivity or a co-cultivation assay. So this essentially is simply that we take our HEC293 detector cells and we mix them with our client's test cells. If our client's test cells have an infectious retrovirus, it will get into the HEC293 cells, and we will amplify that over a minimum of 5 passages and then perform an F-PERT endpoint. And this was give us an indication whether or not we had an infectious retrovirus. So summarizing what I've talked about, we can look for markers of infection. The PCR-based methods are very powerful because they don't require the virus to grow in culture. And that's really important because many virus families can't be grown in culture. However, if you get a PCR positive result, you don't know whether you have an infectious or a non-infectious virus. You can use electron microscopy that Ewan's going to talk about as a general screen, and also PCR. But obviously for PCR, it requires that you know the sequence you're looking for. If you want to look for infectious virus, the only way to do that is in a culture system. And the endpoints can be general, looking for things like cytopathic effect or hemoabsorption, or they can be specific if you're looking for specific contaminants, for example, using immunofluorescence. So finishing off, it's important to remember that every assay has a limit of detection. You clearly cannot test all of a product. So if they have a low-level contaminant and you're testing a small amount of material, there's always a statistical chance you're going to miss it. Therefore, you cannot test for all viruses. So we can never prove the absence of an infectious agent, which is why we have various methods and a holistic testing approach. So all we can ever say to applying for a specific assay is that it was valid, the negative controls were negative, the positives were positive, and your material was negative in that test. But that doesn't necessarily mean there's nothing in it. So it is a risk-based approach. It's a combination of approaches, and it depends on the production parameters, a viral risk assessment of the raw materials, and also the clinical application. So I'm just going to quickly finish off showing you the main regulations I've talked about. So within ICH is Q5A, which is viral safety, which is revision 2 is under consultation. Within the US, although these are old guidances, the points to consider from 93 and 97 set the scene for biosafety testing. I've mentioned a couple of times the FDA vaccine guidelines from 2010, and I've also mentioned the testing for bovine and porcine viruses, which is under the 9 CFR, the Code of Federal Regulations. Within Europe, there are several guidances and regulations under the European Pharmacopoeia. So the main ones I've talked about today are 2616, tests for extraneous agents, and 5.2.3 cell substrates, and 5.1.7 viral safety, which is essentially the same as ICH Q5A.